So we're here today with Dr. Patricia LaRusso from Yale Cancer Center, where she runs the phase 1 trial unit there and also does a lot of work in the thoracic oncology space. And Dr. Fabrice Barlesi, a real expert in immunology at Institut Gustave Roussy in France, who also does a lot of the early stage trials as well as thoracic oncology there. And would be great if each of you could just briefly introduce yourself for the folks that we have on the line here. Maybe starting with you, Dr. LaRusso.
Yes, first of all, thank you all this afternoon for attending, or this evening, depending on where you're calling in from. As Faisal said, my name is Pat LaRusso. I'm a medical oncologist at Yale University. I run their early phase clinical trials program. Slight correction, I don't focus in thoracic. I do a lot more with women's cancers and early phase clinical trials, but primarily solid tumors. Hematologic phase 1 trials are done by another team at my site.
And I have also to thank you for joining this webinar today. It's a great pleasure to share this presentation with Dr. LaRusso. And I'm a thoracic oncologist. I was formerly in the south of France in Aix-Marseille, and joined Gustave Roussy earlier in January, where I'm leading the medical affairs, but also the clinical research. My main expert field is in thoracic oncology, and I'm participating to immunology and early phase programs as well.
Thank you both. So I'll just start by introducing the kind of flow of what we're planning to go through today. So we'll start with just some opening remarks on the next wave of cancer care, what we mean when we say the next wave and things coming through the pipeline in the next generation of oncology therapies. And then we have a few topics that we'll go into in more detail. Dr. LaRusso and Dr. Barlesi will go through some of the data that we heard, and then we'll have a little bit of a discussion and Q&A. We'll take questions off of the Zoom webinar as well. The panelists will ask questions of each other, and we have a few questions teed up as well. And then at the end, we'll close out with some open time for Q&A, and then just some closing remarks on where the field is heading as a whole. So just to kick us off, we wanted to just introduce this concept of what we mean when we say the next wave of oncology or oncology for the future. And really the way that we've been thinking about it at Axiom is across these eight categories broadly, where there's this whole idea of novel IO targets beyond PD-1 or PD-L1. There's novel molecular targets, things like KRAS, that are very exciting lately. Antibody drug conjugates that we've seen amazing data for and also seen in the news lately. Biomarkers and precision medicine, how can we get to the next level of patient selection and really kind of getting a tailor-made approach for every single patient and the individual disease that they have. Anti-cancer vaccines, novel engineered proteins, how can we adjust molecules that we have using these amazing engineering-based approaches, fusion proteins, etc. Similar to that, bispecific approaches. We have antibodies that can do much more than the antibodies of years ago could do. And then cell therapy as well, using cells and engineering them to really kind of have a tailor-made cancer therapeutic. And at ESMO 2020, we saw amazing advances across all of these categories. In terms of novel IO targets, there was TIGIT, there was anti-myeloid approaches like ILT4, other checkpoints like LAG3 and TIM3. I mentioned KRAS and novel molecular targets. Dr. Barlesi presented amazing data from an initiative that he was chairing up called the Pioneer Study, looking at biomarkers. Antibody drug conjugates, I mean, that list is getting longer and longer. Even cancer vaccines, there's an interesting IDO peptide approach from a group in Europe. IL-2 was interesting on novel engineered proteins, next wave of cell therapy, and then a bunch of bispecifics as well. So given everything that is happening here, unfortunately, we had to make the tough choice of kind of selecting out a few topics to discuss. So we tried to kind of balance between what we saw a lot in the news, what we saw a lot of clinicians and industry folks that we speak to discussing, and also what Drs. LaRusso and Barlesi have some experience and expertise in, and really tried to do a distillation down to 4 topics that we'll spend a little bit of time on each of these today. We'll start with TIGIT. This is an area that is getting a lot of discussion, some interesting data, and rapidly advancing in the clinic. So let's take a step back and talk about what we have there. ADCs as well. There's some that are approved and in use in clinical practice, and even more that are in development as well. novel targets, things like KRAS, things like MET that we've maybe known about for a while, but we're just getting to a next level of being able to hit these targets. And then lastly, novel technology. So the ability to have novel engineered approaches like IL-2s or bispecifics. So we'll go through each of these, have a little bit of a data overview, and then discussion and Q&A. For those on the Zoom webinar, feel free to send in your questions as well. And with no further ado, I'll turn it over to the real experts. Dr. LaRusso will start with just talking about TIGIT as a mechanism, and then the data that we saw for an agent called vibostolimab. So, Dr. Russo, what is this TIGIT?
Yeah, so TIGIT actually stands for T-cell immunoreceptor with Ig and ITIM domains. It's actually a transmembrane inhibitory receptor on both T and NK cells, which regulates immunity via the CD226 TIGIT PVR pathway. So as you can see, TIGIT is important in CD8 positive T cells as well as NK cells, and also, as you can see, dendritic cells. Whereas on the CD8 positive T cells, it leads to suppression of the T cell mediated killing and inhibition of CD8 positive T cell priming and differentiation. So it's more of an inhibitory effect, as well as on NK cells, leading to suppression of NK cell mediated killing. And then on dendritic cells, induction of the dendritic cells towards tolerogenic activity. Next slide. So there are several TIGIT focused or targeted agents in clinical development, and there were at least 4 presentations, either by oral presentation or abstract, that were presented, 2 of them actually being trials in progress at this most recent ESMO meeting a few weeks ago. I'd like to spend a few minutes on vibostolimab, which was presented in both PD-L1 naive as well as PD-L1 refractory non-small cell lung cancer. Let's start with the PD-L1 naive study that was presented. This is a compound from Merck. It was a phase 1 clinical trial that was done in IO naive non-small cell lung cancer patients. Phase 1 trial, so not a huge patient population. There were 41 patients across multiple lines that had multiple lines of therapy, but were PD-L1 naive. 27% of the patients were actually treatment naive for advanced disease. And the PDL status was only known in 25 of the 41 patients. And of those 25, there were only 13 that had a TPS score greater than or equal to 1%. And 12 of them were less than 1%. And the monoclonal antibody they'd used for the TPS testing was 22C3. In terms of efficacy in this immuno-naive population, they had a confirmed response rate of 24%, which included 9 PRs and 9 partial responses and 1 complete response. And there were an additional 2 unconfirmed partial responses that were also observed, to give an overall response rate in the confirmed and unconfirmed population of 29%. The median progression-free survival was 5.4 months. And if you looked at the TPS greater than or equal to 1%, the response rate was 31%. There were 4 patients, compared to 25% or 3 patients in those who had a TPS score that was less than 1%. It was relatively well tolerated. And the company is planned to move forward, according to the presentation, with a phase 3 trial of vibostolimab plus pembrolizumab in 2021. But as you can see in the bottom right, the waterfall plot, as you can see, there were several responses, many not hitting that 30% target, but nonetheless, somewhat encouraging data in this non-IO population. Next slide. Then they also looked at the same drug in PD-L1 refractory patients. Again, 41 patients were enrolled with the monotherapy of vibostolimab, and then an additional 38 patients were enrolled with the combination of vibostolimab plus pembrolizumab. So there were about 80 patients, 79 patients total. In these patients, they had previously received at least 2 doses of PD-1 or PD-L1 targeted therapy prior to enrolling onto the trial. But it did not necessarily have to be their most recent therapy for their malignancy. In the monotherapy arm, there was only one confirmed response for an overall response rate of 2%, with 2 additional unconfirmed responses. And then in the combination arm, there were 2 confirmed responses for an overall response rate of 5%. Again, toxicity was as expected, especially with the addition of the pembrolizumab arm. And if you look at the waterfall plot in the bottom right-hand corner of this slide, you don't see as robust of response, but keep in mind that these patients had all been exposed to prior immune checkpoint therapy. Just one other comment to make about the first study that we talked about. Those waterfall plots were more encouraging than this, as you saw. But you also have to remember that what would the response have been in the IO-naive patient population if we had even gone one step further, or the study had gone one step further and only enrolled or enlisted frontline metastatic patients. Many of those other patients had been heavily pretreated, relatively heavily pretreated. Next slide. And so, as I told you, there were other TIGIT-targeted drugs that were presented. These were both trials in progress TIP studies that were presented. There was no data, only essentially the trial design. But interesting, these are 2 somewhat different type of TIGIT-targeted drugs. The Seattle Genetics is an FC-enhanced anti-TIGIT monoclonal antibody, and they're looking at their compound both as monotherapy and in combination with pembrolizumab in 3 distinct tumor baskets. And the patients had to have been refractory to all available therapeutic options to be recruited to this trial. And then Arcus had a very interesting 3-drug combination, and their TIGIT monoclonal antibody is actually an FC-silent monoclonal antibody. And they were using it in combination with their anti-PD-1 compound, in combination as well with the 3rd drug, the A2AR compound. And interesting, this study is in frontline non-small cell lung cancer patients, and they're pre-selecting to have a PD-L1 expression in their patient population of at least 50%. And as you can see, that the arm 1 was monotherapy, arm 2 was with Dumvalatinib, and arm 3 was the 3-drug combination, as I had discussed. And with all of these 3 arms, they are looking at PFS as well as overall response rates, but a 3-arm phase 2 clinical trial. And as you can see on the right, there are many other TIGIT compounds by many other companies that are in clinical development. As you can see, anywhere from phase 1 or IND only through phase 2 and phase 3 trials. So Fabrice, I've got a quick question to ask you regarding this data. This is all TIGIT, and especially with the first compound, the Merck compound, it's actually being looked at in the lung cancer landscape. What do you think about that data with either the IO naive or the IO previous treated patients in looking in the non-small cell lung cancer landscape, being a thoracic oncologist yourself?
I think that all the data we have seen so far with the TIGIT inhibitors are very encouraging, I would say, especially in the naive patients landscape. We see, and it is exemplified in the study you just presented, we see quite encouraging response rates, even if some of them are unconfined, but we are close to the 40% response rate, which is something that is very interesting. The only drawback of all of this study, at least for the moment, is the fact that we are globally unable to predict in what kind of population of patients it will be the better place to be used. It seems to be quite closely related to the PD-L1 expression, and we know that there is a concomitant, usually expression of PD-L1 and TIGIT. then it is not surprising that this kind of combination work better in PD-L1 positive patients. And it is exemplified with the tiragolumab, but also exemplified with this new data presented with the Merck compound. But I would say that we probably have to a little bit understand where we should dose this drug, and especially when we look at those patients treated in PD-L1 refractory patients, probably because we have to better understand why they are refractory. Probably we have also to better classify those patients. Would it be a primary resistance to the immunotherapy, then patients progressing very early after starting immunotherapy, or patients progressing after some response or stabilized disease? Then probably it's two different patients populations with probably two different types of mechanism of resistances. And probably if we look just at the PD-L1, we have, I would say, we suggest in the Pioneer trial that there could be an increase in the PD-1 or PD-L1 expression once patients are on the drug. Then maybe it could be a drug that could be used in addition just for the patient, maybe with just a stable disease as an example or something like that. Then I think we have to a little bit better understand, but of course we are, I would say, quite at the beginning of the development of those drugs. It would be important to have a very strong translational research along with the clinical development. And I think that it's probably one of the lessons we may derive from the Merck data just presented at ESMO, the fact that they don't have even the PD-L1 status for a large part of the patients. Then we need to collect tissue for all of these patients, probably to try to rebiopsy those patients who will progress quite early, just to better understand the concept. I think the last study you presented, the study from Arcus, is also very interesting when we look at all the different arms of the study. One with just the TIGIT inhibitor alone, one with the combination of the PD-1, and then they added, because they believe that, they showed that there is an increase in the adenosine pathway, which is an immunosuppressive pathway. Then they add an adenosine inhibitors, which is, I would say, very smart regarding the development and the different arms. We will see if it translates in a clinical benefits, but I think it's we are going to see more and more very nicely designed study, and it's very, very interesting to see for all of us and our patients.
Do you think we'll ever get to the point where we're going to do personalized immuno-oncology trials as well, based on all of these selective targets that are now either, or these drugs that are either enhancing various components of the immunologic pathway or suppressing them?
Clearly, I believe there is no other way to go forward. It will be probably more difficult than the experience we have with targeted agents, probably because the immune environment is very complex, and it's probably also related to host factors. But clearly, I think that the future will be based on precision immuno-oncology. We have to learn a lot. It's not so easy. In the Pioneer study, we had the possibility to rebiopsy the patients only in 2/3 of the cases, then it's not so easy to do that. But I think that we learn a lot of all of these ongoing trials. Also, I think what we will get from the liquid biopsies will also help us to better understand how those drugs are working. Then I clearly believe that in a few years from now, we'll be able to better design and better drive the clinical decision based on biomarkers.
It's interesting, Dr. LaRusso. It almost seems like what you might even be describing and advocating for. It's almost like a lung map or NCI match type of a protocol, but for precision immuno-oncology is kind of the level of biomarker sophistication we need to get towards.
And I believe the NCI is actually working towards an immuno-matched type of trial, as well as a molecularly targeted combination trial that they're going to have many arms. So that, I think, is also going to be informative, especially if, like Fabrice had said, we do get those serial biopsies to help us understand, partnered with liquid biopsies, so that we can determine whether or not we can do non-invasive profiling and follow these patients that way to determine when, hopefully, we can interject with additional drug combinations or add additional drugs to previously ongoing therapeutic interventions.
Yeah, definitely. And also one question I wanted to pose to both of you is, it was interesting on that last slide that you had with the 2 trial in progress posters. One poster described their antibody as an FC enhanced approach. One poster described their antibody as an FC silenced approach between the 2 different ones. So I guess I'm just curious. I know there's been some different preclinical data that's been shown on this, nothing really in the clinic, but what do you think about this whole FC debate when it comes to the TIGIT antibodies? Will this matter? And are there other analogs for this situation and other antibody targets?
I think I'll start with that, Fabrice, and then you can chime in. We cure a lot of mice. We see a lot of positive data in mice that we unfortunately don't translate to patients. Otherwise, we probably could have been able to eradicate cancer a long time ago. So there are, preclinically, at least in the lab, they're showing differential benefits, not only in terms of efficacy, but potentially also in terms of toxicity as well. But I think the real answer will come when these trials are much more developed and we start seeing differentials potentially in terms of both efficacy and toxicity. I don't know about you, Fabrice, but I don't know that we can answer that right now in the clinical scenario because the data is not yet mature in those clinical trials. Fabrice, do you have anything to add?
No, I have no, I would say, specific data to add to what you say. I think it's I would say within the development of the drug, the chemists and the developers found one of the, I'm not sure that it is driven by the theory. I think it's probably more what they say when they are developing the drugs. And but I think I'm not sure that it's the main point that will drive the benefit for these drugs if we are not able to select the patients on the basis on what happens in the tumor. I think it will be maybe it will come in addition, maybe to differentiate 2 different drugs, but the patient selection will be the most important things to do.
And unless the differential is huge, Faisal, it's going to be very difficult because, it's hard to put drugs like this head to head that have similar mechanisms of action into one clinical trial, especially when you're dealing with multiple different pharmaceutical sponsors. And you're dealing this early on in the development of these targeted agents. Unless there's an earth-shattering or a real significant differential in a very similar population in 2 separate trials, it's going to be very difficult initially to answer that, I believe.
Really?
Yeah, it almost reminds me of how many years have we been talking about PD-1 versus PD-L1? It's almost a political debate at this point, but we won't get into that.
Yeah, I don't know that. Yes.
Okay,
Yes.
Great. So then maybe we'll switch gears a bit then and go into our next topic of antibody drug conjugates. I mean, this is an area that has been around for a bit, but recently there's been just a acceleration of excitement on this with recent data, a lot of more molecules entering the clinic, being approved. So maybe we'll start with just an overview of what do we mean when we talk about ADCs, how do they sort of work, and then we'll go into 3 different targets and their data. We'll go into TROP2, we'll go into HER3, and then we'll go into NaPi2b. So how about we'll start on just the mechanism with Dr. LaRusso.
Okay, thank you. Thank you very much, Faisal, for introducing that topic. So yeah, antibody drug conjugates, I think, are becoming more and more exciting. And I think a lot of reasons for the increased excitement is as the science matures and we identify targets that are meaningful, but it may be difficult potentially to deliver a drug any other way. An antibody drug conjugate with that monoclonal antibody targeting that specific target, for instance, a receptor on a tumor cell, and being able to deliver a cytotoxic agent by using that monoclonal antibody is really exciting. So we use the antibody as like the driver. It gives us the roadmap to get to the tumor cell, but the linker is extremely important as well. So the antibody drug conjugate is really composed of 3 parts: the monoclonal antibody, the linker, which links the cytotoxic agent to the monoclonal antibody that will hopefully eventually be delivered to the cancer cell, and then the cytotoxic agent itself, which hopefully will go intracellularly and be able to kill that tumor cell. The linker is, the compounds are usually delivered intravenously, obviously, for most, all of them that I know of at least. And then once they get intratumorally, they're broken down by the lysosome, which obviously takes the linker off the monoclonal antibody and allows the cytotoxic agent to get intracellularly, and then externally, the cytosolic drug leads to cell death, and then you get external drug release. I think one of the other things that's really exciting about antibody drug conjugates in 2020, and we saw this with data that was presented either orally or by poster at ESMO, is not only the new targeted agents, the new monoclonals, but also the new payloads or the new cytotoxic agents that now allow us to drive home drugs that maybe may have been too toxic initially to give by themselves, or had to be metabolized previously into an active moiety. And as a result, we could never get enough of that active moiety intracellularly to get a real cytotoxic effect. So if you go to the next slide, I think a good example of this is the TROP2 data that was presented, sacituzumab govitecan. And there were two presentations, one in triple-negative breast cancer and one in urothelial breast cancer, and urothelial cancer, excuse me. And, it's targeting a novel target, TROP2, and it's delivering SN38. And, SN38, obviously, we all know is the active moiety of irinotecan. And in both of these tumors, we have seen previously response with irinotecan. But in fact, there was even an oral irinotecan that had been developed many years ago and was looked at in breast cancer, but was never further developed because it was always really hard based on the toxicity of the parent to get that active metabolite delivered in sufficient quantities to give us efficacy without toxicity. So ASCENT is looking at sacituzumab govitecan in triple-negative breast cancer in relatively heavily pretreated patients. It was 3 or more lines of therapy, and I believe a median in this study was a median of 4. And it was randomized 1-to-1, 529 patients to either sacituzumab or physician's choice. And there were 4 active drugs, 4 drugs that are used in the advanced setting: eribulin, capecitabine, navalbine, and one other drug that were in the physician's choice portfolio. But as you can see, there was significant efficacy in this randomized trial. Not only in, remember, heavily pretreated patients, median PFS differential of 5.6 versus 1.7 months. That hazard ratio for both progression-free survival as well as overall survival is quite impressive, quite impressive. And the survival differential between the TROP2 targeted TROP2 targeted ADC versus physician's choice was twofold, a median of 12 versus, well, just under twofold, a median 12.1 versus 6.7 months differential. And you can see in the bottom left, the overall survival Kaplan-Meier curve. That curve separates very early and is pronounced through the entire follow-up, as you can see here, out to about 2 years for both arms. Quite impressive. This p-value is 0.0001. A novel, very well tolerated, as you can see, looking at safety, antibody drug conjugate for this patient population that really doesn't have a lot in this setting. So this is going to be a major advance for our triple-negative breast cancer patients, or our patients with triple-negative disease. But it's not only a drug it looks for this patient population, because if you look at the TROPHY study, and this was a single-arm study of sacituzumab govitecan in heavily pretreated refractory metastatic urothelial cancer, 113 patients that had seen at minimum a platinum and an immune checkpoint in, therapy prior to coming on this study. and there's a median of 3 prior treatments in the metastatic setting. The response rate, again, is 27%, with a median duration of response close to 6 months, and a median PFS of over 5 months, with a median overall survival of 10.5 months. And I think just looking at that waterfall plot, if you're a visual person, that waterfall plot, I think, really speaks for itself, demonstrating therapeutic advantage, not only in triple negative, as we saw on the left, but also in metastatic urothelial cancer. And because TROP2 is expressed, well, actually overexpressed, the trophoblast antigen 2 is overexpressed in many different tumor types, as well as solid tumors and hematologic malignancies. I think the future for this compound is really quite exciting, and hopefully will lend therapeutic advantage for multiple different tumor types to give this advantage to many different patients with many different tumor types in the future. Next slide.
Yeah, that's a great point you make, Dr. Russo. I mean, the visuals are really just stunning on this Kaplan-Meier and the waterfall plot here. So the next target we're going to discuss is a HER3 ADC. This one's called patritumab deruxtecan. This is specific to the EGFR mutant segment of non-small cell lung cancer.
Right. And Fabrice, do you want me to start with this? Do you want to take this? Because I know you're a lung cancer expert.
Yeah, but yes, start. No problem.
I'm sorry?
You can start.
Okay,
Yes.
yeah.
Yeah, sure.
So yeah, you know, I've been doing drug development for many years, and I've dealt with, or I've done phase 1 clinical trials with monoclonal antibodies only, not antibody drug conjugates, that targeted only HER3. And unfortunately, just delivering the monoclonal antibody didn't really give us, in several phase 1 clinical trials, we didn't really see many signals of activity with the monoclonal antibody just targeting HER3. So adding a payload to a compound, like to a monoclonal like HER3, as we're doing with deruxtecan in this antibody drug conjugate, I think has actually been very exciting. This is a study that was done in EGFR mutant non-small cell lung cancer. 100% of these patients had been previously exposed to EGFR tyrosine kinase inhibitors. Close to at least 80%, if not more, had also seen osimertinib as one of the EGFR TKIs. And yet, if you look at the waterfall plots here in the bottom right-hand corner, you see that delivering this HER3 targeted antibody drug conjugate, so hitting the EGFR family, but then allowing the payload to go intracellularly and deliver the cytotoxic agent, actually is another way of manipulating the EGFR target to be able to get anti-tumor activity potentially. But this was a phase 1 trial. And there were 45 patients that were enrolled on this trial. All had to have EGFR mutation, and all had to have at least one EGFR tyrosine kinase inhibitor and at least one line of platinum-based chemotherapy to go on this study. These were heavily pretreated patients. Despite the heavily pretreatment of these patients, the overall response rate was 25%. You don't see this in this patient population typically with standard chemotherapy. This is quite impressive. And the disease control rate was 70%. So in this phase 1 dose escalation trial, they were seeing significant responses using this HER3 targeted ADC in a select EGFR mutant population that had been resistant or refractory to small molecule tyrosine kinase inhibitors. This waterfall plot in the bottom right-hand corner just, I think, says it all. I mean, this visual is quite impressive, given that this was a phase 1 dose escalation trial. Fabrice, do you have anything to add? What do you think of this data?
You're right. It's a very important data set. And I think, as you know, when we have exhausted EGFR TKIs and platinum-based chemotherapy, the place of the immunotherapy is clearly not clear enough in this patient's population, then the availability of further line of treatment is very important. I think it's, for me, it's both very appealing data because it highlights the fact that chemotherapy may still have a place, but this chemotherapy should be driven by a target, and we know that the expression, the overexpression of R2 or R3 in this patient's population is important. But I think it's just also maybe the first step, because probably one part of the toxicity we may have seen in these patients' populations, as you said, heavily pretreated, may be also the consequence or driven by the platinum-based chemotherapy they received before. Then maybe if we are able to use this drug earlier in the management of those patients, we may have a better safety profile. The second point is, of course, it's a data escalation, then it's monotherapy. But we know that in this patient's population, there is probably a rationale to try to keep the EGFR blockade in this patient's population and then add something else in order to overcome the mechanism of resistance, which may be linked within the ER pathway by the expression of 2 or 3. Then I think that it's probably, I'm really very interested to see the data with the combination of an EGFR TKI. That will be very, very important. Of course, we just have in mind what how we need to very carefully look at ILD in this patient's population when combining these antibody drug conjugates with EGFR TKI. We had some alerts in the past when doing this, when doing other types of combination, that we will have to follow those patients very carefully.
Yeah, I was going to ask you, Fabrice, what do you think? I mean, this is a dose escalation, and just in this 45 patient, or just in this patient population, this was an expansion to the phase 1 trial as well, that they did report on. So, I apologize that I was talking primarily about escalation. A lot of the data was also included in an expansion cohort of patients. But 3 ILDs and 9% discontinuation as a result of adverse events. What do you think of that? What's your take on that, Fabrice? Because you deal with lung cancer all the time.
Yeah, you're right. But I think, it's difficult. We need to see all these ILD events very carefully. Those patients receive any previous radiotherapy, as an example? Do they receive any previous immunotherapy as the previous lines of treatment? And we know that there could be some, we had in the past some adverse events in this when trying to combine immunotherapy agents with EGFR TKI as an example. Then I think that it's not very important. It could be seen, but when we look at the data we have with other kind of treatments, the rate of ILD is usually lower. We need also to look. I don't have in mind where this study was conducted, but we know that there could be a little higher susceptibility of Asian patients regarding ILD compared to the Caucasian patients. Then maybe we have to look at all these points. The discontinuation rate is probably linked to the grade 3 adverse event we have seen. 1/3 of thrombocytopenia, some severe neutropenia, but we know that it could have been influenced by the fact that those patients have been heavily pretreated before. Then I think that probably in the future, the use of these antibody drug conjugates will come probably earlier than it is right now.
Yeah, there were a significant number of co-authors on the oral presentation that were from Asian sites.
Yeah.
So that may have skewed the data. Yeah. Also, I mean, many of these patients, I've seen ground glass not uncommonly with drugs such as, gemcitabine, and we don't know what a previous ground glass or low-grade ILD what impact it would have when you're giving a drug such as this that also causes ILD. So, I think, do you think that moving this drug earlier into the continuum could help alleviate some of that toxicity?
Probably we may imagine that the safety profile will be better if we use this drug earlier. It's likely, I would say, but we have to see. I think it's the same as we said before. We have also to see where this drug will be better placed compared to where we'll place the immunotherapy in this patient's population. Will it be with the chemotherapy? Will it be separately? Will it be combination of immunotherapy? I think there are a lot of open questions in the management of these EGFR mutated patients resisting and of EGFR TKI available to date.
Yeah, and I think that's where some of those next steps will really bring some more clarity to the discussion, where there's, you know, phase 2 looking kind of more cleanly at that patient population that they did the dose expansion, and then combination is an interesting approach that they're, I believe, just going to think about starting in phase 1, which will be interesting to see, like you said, Dr. Berlese, keeping that that EGFR inhibition, kind of keeping that pedal on the gas, if you would, and then adding in the HER3 cytotoxic. In the interest of time, I'd like to just kind of shuttle us forward a little bit to this kind of other target here, the NaPi2b, which it was kind of interesting, Dr. LaRusso, you were mentioning how with HER3, we kind of went from the sort of naked antibody not working out, but then these ADC approaches looking much better. Interesting story on NaPi2b. There was a kind of more elementary antibody drug conjugate a few years ago that didn't quite work out from a different company. I believe it was Genentech. And then now this is a small biotech that has you know, a slightly better ADC construct. And Dr. Berlese, why don't you tell us a little bit about this data and what we're seeing here?
I think it's also a quite interesting target because it's then NaPi2b is a sodium-dependent phosphate transport protein, which is broadly expressed in solid tumors, and especially in epithelial ovarian cancer, but also some other tumor types as non-small cell lung cancer. And in this study, what makes a small difference with the data we have just seen before is the fact that they were able to separate the patients in different populations on the basis of an immunohistochemistry using a H-score for NaPi2b. And they have then two different populations, approximately the half of the patients with an high score and 1/3 with a low score, and 15% with for whom the score was not known. Then they enrolled in the study 47 patients. And as you can see, those patients are also heavily pretreated ovarian cancer patients, platinum resistance, and you can see that the vast majority has several prior line of therapy in this setting. But in spite of this, the number of treatment received before, the response rate was 34%. When the response rate in this patient's population with a drug that are usually used is closer to 5 to 10%, then clearly it's clinically meaningful results. Even, of course, if it's a small study, it's a small sample size, it needs to be confined, but it's really, really encouraging. When we look at the type of response, they got even 2 complete response in this patient's population, and almost 3 complete response when you look at the waterfall plot on the right. Then the second point is that, and the good news is that there is I would say a sign of potential patient selection regarding the use of this H-score. It seems that the probability of response is a little bit higher when this H-score is higher. Then it's something that is interesting. And the second point that is very interesting is the fact that this response seems to last for a long time, and the duration of response was not reached when for the data presented in this high subgroup patients. There were no grade 3 treatment-related adverse events. The adverse events otherwise were as expected for this kind of drugs, related to asthenia, GI events, liver enzymes increase, and some hematological adverse events like thrombocytopenia and neutropenia. As you can see, in some patients, the dose reduction was mandatory, and some has to discontinue the drug. And what is important, I think, when we are discussing these patients with a disease with, unfortunately, a poor prognosis, is the fact that this drug will hopefully become very quickly available, and the drug got FDA fast track designation which is something that highlights the fact that when you are designing a drug with a very good rationale, you are conducting a trial with I would say a design that takes into account the probability of the presence of the target, then you may got very important, clinically important results for patients. And I think it's something that is highlighted by the results presented here. I don't know,
I forgot, what was the payload? What was the payload of this drug? Do you remember, Fabrice?
Yes, it's auristatin.
Ah, another mitotic poison. And even then, you still got a great response. That's beautiful.
Yeah. What do
So
you think?
We had a,
you know, you are an expert in women's cancer. Then what do you think, Pat, from these results?
I mean, if we can reproduce this in a larger trial, this is very encouraging data. I see a lot of these patients. There's not a lot of great therapies for them, especially many of these patients had had at least 4 lines of prior treatment. This is extremely encouraging data.
Great. So wanted to shift to the questions a bit. I think we might have addressed this first one on AEs a bit. Thanks for those comments on the ILD especially. What was interesting, Dr. Berlese, I know you're, there's no bigger proponent of biomarkers than you. It was interesting on that last one that we looked at. They were looking at the IHC for the antigen that they're directing the cytotoxic towards, but for the first two, there wasn't any IHC for the antigen. So I'm just curious, especially in that phase 3 study, Dr. Russo, that you went over, how are they seeing such a broad efficacy when you would think that if you're targeting the cytotoxic towards an antigen, would you not want to select for the patients with tumors that express that antigen? How are we getting an all-comers type of efficacy?
Yeah, you're talking about the ASCENT trial?
Yeah.
Yeah, so I think one of the advantages they had, but I would still like to see the biomarker data, and I'm hoping that they did collect that data, at least with archival tissue. And Fabrice, please correct me if I'm wrong, but it is encouraging data, but I would love to see the biomarker data. I would like to see the TROP2 expression relative to response in that randomized phase 3 trial. When you have a targeted drug like that, even if you're getting a really, really good response, we did see some toxicities, and there may be other drugs that come down the pipeline that are better drugs in the future. And so I think it's important, and I think we're obligated to look at least at the proposed biomarker, which is TROP2, and see if we're seeing a significant differential. Even if there's a much smaller population that isn't expressing TROP2 relative to the population that is, we need to know what the differential of efficacy is, I believe, in those two populations as we're moving the drug forward. And I would think that many regulatory agencies, when you have a designated target that you're going after, even in an ADC, would want to know what the expression level is relative to efficacy. Fabrice, what do you think?
I think to answer the point of Faisal, the fact also that we have to face some heterogeneity in the tumor, then it's sometimes difficult to assess the presence of the target on a small biopsy. Maybe the target could be present elsewhere in the other part of the tumor, in some metastasis, then explaining why in some cases the drug works without the identification of the target at this place. I think, of course, I do not foresee a large future for drug that will not be developed, at least with a potential biomarker. I would say just a potential help for the clinician to select across different drugs. We are just at the beginning of the development of all those drugs. How we will choose in the future when we will have 1, 2, 3, 4 different possibilities for one patient. Then if we don't have the help of a biomarker, it will not be possible. But a biomarker does not mean specifically a tissue biomarker. It could be something in the blood. It could be also something with the metabolic images. We made imaging tracers, metabolic tracers that will that will help us to identify the presence of the target, as we are doing in some neuroendocrine tumors before using some drugs. Then we may imagine that in the future, metabolic images will help us to identify the presence of the targets in the vast majority of the the tumor and the metastasis, and then use this drug driven by this by the presence of this the target defined by the imaging techniques.
Got it. Very interesting. And then I guess this last question here was, we saw the HER3 approach, the TROP2, the NaPi2b. Are there other antigens that are well characterized, known expression in certain tumor types that it can also benefit from this ADC type approach? When I wrote this question, I was trying to think about other EGFR resistance mechanisms even. I know there's MET there, can even just hit EGFR itself. I saw there's a couple in development. What other targets could be out there that you've either heard about or you think could benefit from this type of an approach?
I think as the science unfolds, we're going to see a lot of other targets. Nectin-4, for instance, is one that's been, developed, and you've seen the data. It's good, and that's another one of these targets that they're developing. LIV-1 is another one. So I think we're going to be seeing a lot of these tumor antigens as we start, because the science, I think, is driving us to be able to do this. Fabrice?
We had also this the mesothelin, which was a little bit disappointing, but I say very encouraging results have been seen also with CEACAM5. We presented the data at ASCO, and for high expressor for CEACAM5, there was very interesting results in non-small cell cancer patients, and phase 3 is going to start. Then I think we have, I would say, a large portfolio of potential targets. But of course, I think as Dr. Lorusso very nicely exemplified at the beginning of this part, it's not related only to the target. It's related to the antibody. It's related to the payload. It's related to the linker. Then I think there are a lot of aspects that may influence the final results you get. Then, of course, it's a large space for research.
I think one of the advantages of the TROP2 and the HER3 is the payload is unique relative to a lot of the payloads that had previously been investigated, and some which were actually FDA approved. That I think that makes a huge difference. I think that's very important, just like Fabrice said.
Yeah, definitely. And even one of the advantages of this ADC approach, you went over the mechanism of it, Dr. Russo, is you don't even necessarily need the tumor to be driven necessarily through the antigen. It just needs to be expressed there, and ideally uniquely expressed there relative to the surrounding tissue. So that's potentially one advantage from a drug development perspective.
Yeah, I mean, HER3, you're right on. HER3 is a perfect example. I've developed a few of those monoclonals, and we didn't get much signal of activity, but the ADC has been very telling.
Exactly. So let's go to our next topic. Then we have KRAS and novel targets. So we'll start on KRAS, then we'll go to an EGFR MET approach. So we'll start with KRAS and Dr. Russo, and then we'll have Professor Barlesi talk about the EGFR MET. And I'll just add that we're actually quite honored to have Dr. Russo discussing this, because she had the honor of writing about this in the New England Journal. So we're getting some next level expert opinion here.
Yeah, so this is a pretty exciting drug because we know for many decades, at least many years, people have tried to target KRAS, and it's been a very, very difficult, target to drug up till recently. I mean, recently, I think we're seeing the change. This is some of the data, the preliminary data, that is showing that, Kevin Shokot really is has paved the way, and many others after him, and many working side by side in terms of developing the right chemistry to be able to target this compound, this target. This is a specific mutation, G12C, KRAS G12C, and it's the Amgen 510, or the sotorasib small molecule that was recently in phase 1 trials presented by David Hong, most recently at ESMO, but there have been other oral presentations of this compound. I think the future is pretty exciting. I think we're going to start seeing other KRAS allelic targets. I think G12D is right on the, the tail of the G12C story. This was a phase 1 trial, and it was a dose escalation in advanced solid tumors. And then what they did is they did 2 major expansions. They did an expansion all in KRAS G12C targeted tumors, but they did an expansion in non-small cell lung cancer as well as colorectal cancer. They had 59 non-small cell cancer patients that they reported on, as you can see here. There were 4 dose cohorts, as you can see, and there was an overall response rate in this phase 1 trial of 32%, with 19 patients having a partial response. But what was really nice is that the disease control rate was 88%. Their recommended phase 2 dose was the highest dose. They didn't see DLTs at that dose, but at that dose of 960 milligrams, they got an overall response rate of 35% with a disease control rate of 91%. As I said, there were no DLTs. They did see a few toxicities. There was some transaminitis. There was some diarrhea, but the discontinuation rate was very low from toxicity. So it's a very well tolerated small molecule. And the phase 1 trial was called CodeBreak 100. And they, as you can see, have other studies that are underway. CodeBreak 200, 101, 105. Many of these looking at select tumor types with various drug combinations. But I think the visuals on the right say it all. Look at that waterfall plot. Look at the vast majority of patients. They may not have gotten that PR, but they are below the line, not above the line. So stable disease with minor regressions. And remember, many of these patients were heavily pretreated, some with IO therapy, platinum-based chemotherapies, et cetera, especially in obviously the lung cancer population. And, the median progression-free survival, although there's no comparator because it was a single-arm study, was 6.3 months, with some patients going out, obviously, as you can see, significantly longer than that. So this is very encouraging. What does it tell us? That, the chemists are starting, they've been working really, really hard, and we can now target KRAS, specifically in this scenario, G12C. There are other compounds that are following right behind that are targeting this drug. Mirati has a compound in clinical trials, and I believe Genentech Roche also has a drug in clinical trials targeting this specific target as well. But I think the future is interesting, not only as monotherapy, but I think that, it may lend itself, at least preclinically, we're seeing preclinically with articles that have just recently come out in the last 4 to 6 to 8 weeks, that there's various combinations that may enhance or may help to increase the efficacy of this drug. Keep in mind that these patients were not frontline metastatic, they were heavily pretreated patients. And unfortunately, we did not see any pharmacodynamic data in this presentation to lend itself to know whether or not we are hitting the target as hard as we can or as hard as we should, but encouraging data nonetheless.
And I guess with KRAS as well, especially just foreshadowing, the next abstract we're going to look at is looking at shutting down EGFR and MET kind of at the same time. So almost like hitting a resistance mechanism before it arises. And KRAS is also interconnected with, different targets, different pathways with the tumor growth. Any comment just on resistance and the combinations and sort of making sure that we're sort of fully shutting down a pathway?
Yeah, so there's a couple really good articles that have come out recently. One by the Mirati team. I think Jamie Christensen was the senior author on that paper, looking at another small molecule that can, they really feel, at least preclinically, has been shown in some of their preclinical models to really help overcome or delay the onset of resistance. And that's a SHP2 targeted agent. I don't know where that is in clinical development yet. But also there have been others that have looked at other compensatory pathways. There's recent publication looking at targeting KRAS and PI3 kinase simultaneously. And because one of the proposed mechanisms is compensatory activation or upregulation of alternative pathways, and PI3 kinase is one of those. Will you be able to combine a drug such as KRAS G12C inhibitor with another small molecule like PI3 kinase? Looking at this toxicity profile, you may be able to get away with it. Will you be able to combine it with immune checkpoint inhibitors? There's data looking at it in combination with immune checkpoint inhibitors preclinically, not necessarily this compound, but other KRAS G12C targeted compounds as well. And it looks like that is, that's a nice combination potentially as well based on preclinical data. And I'm sure people are bringing that into the clinic as well, that combination. But we can't overlook potential mechanisms of resistance with those compensatory pathways. And we have to look at that as we're developing these drugs, not only at baseline, but as we treat these patients. Many of these targets we'll be able to follow with liquid biopsies. I'm sure Fabrice can tell us a lot more about that.
No, you're absolutely right. I think that first, I think for those who are not familiar with lung cancer patient, we just have to replace KRAS mutations are an adverse prognostic factors. For those patients, we know that the standard of care at this time of their treatment would be a monotherapy with a potential response rate around 10%, and it's clearly threefold higher with this drug. What we have to understand better right now is the mechanism of resistance, but I believe that the mechanism of resistance may be driven by the concomitant molecular alteration that we may have in addition to KRAS. And I think we know that those patients are usually EV smokers with a lot of concomitant molecular alteration, and it's made explain the heterogeneity of the responses and the heterogeneity of the duration of the response. But as Patricia Russo said, it's very likely that when you are blocking the MEK pathway, probably the PI3 kinase mTOR pathway is activated as in order to overcome the blockade by the KRAS inhibitors. Then it's probably something we have to explore. It will not be easier with to combine both these kind of drugs, at least with TKIs. But with the progresses we have made in the design of the drugs and the tolerability of the drugs, we may do that. The other point is, when would it be the better place for these drugs? And we all know that when we have an active drugs, and especially an active target therapies, usually it works better when you are using this drug earlier in the treatment of the patients. In addition, we know that those patients are probably usually a little bit more sensitive to PD-1 or PD-L1 inhibition. And it's clear that combining both of these drugs in the early management of those patients would be very, very appealing. And I think that it's one of the setting of the trials that will start very soon, or already started in some countries. Then we have to better understand how to use these drugs, but clearly it will change completely the management of this part of our patient, which represents a non-negligible part, probably around 20% of our patients, up to 20% of our patients, and we will be able to target the G12D with at least drugs are on development, and it will be also something important because it represents also one non-negligible part of our KRAS mutated patients.
Yeah,
and I think one of the things that Fabrice has pointed out is the heterogeneity, because not presented in this presentation, but included in the New England Journal article from Hong and colleagues, is also the colorectal data. They did an expansion cohort in colorectal. They did not see the same disease control rate in colorectal at the same dose of 960 milligrams as they did in lung cancer. The disease control rate was quite a bit, was quite a bit less. And, they're both very heterogeneous tumors. And, unfortunately, we don't have molecular data from these trials yet I'm hoping that they did collect molecular data on these patients, and we'll see this data over time, hopefully soon. But the fact that the differential in control rate was there between colon and colorectal cancer and non-small cell lung cancer, you're probably dealing with a different set of mutational aberrations in addition to KRAS in these two distinct histologic subtypes of disease.
Right, you really get the sense that with this KRAS, you mentioned the medicinal chemistry breakthrough that we had here, but really get the sense that we're at the tip of the iceberg, or the tip of the tip. There's a lot to go on this KRAS story, so we'll stay tuned.
I always tell my house staff, all that is BRAF is not created equal, because BRAF colorectal response totally different to BRAF inhibitors than BRAF, positive melanoma, BRAF mutated melanoma. We're probably going to see the same thing here, but with a better monotherapy benefits.
Yeah, exactly. Next one we're going to go here. This was presented by Dr. Byung-Chul Cho from Korea. Very interesting approach. It was an EGFR MET bispecific called Amiventumab, a third generation EGFR TKI called lazertinib in two distinct EGFR mutant lung cancer populations. Dr. Borlesi, walk us through what we're seeing here, because there's quite an interesting looking waterfall plot that we have on the page.
Yes, it's a very interesting study and a very interesting drug. We know that the MET amplification is probably the main resistance mechanism, both when using first and second generation of EGFR TKI and even the third generation EGFR TKI osimertinib. Then, based on the principle that you may overcome resistances in targeting them very early, the concept to try to block both EGFR and MET is very interesting. We have some data presented with the combination of two different TKIs, and this study and this compound acts a little bit differently. We have a bispecific monoclonal antibody, which is targeting EGFR and MET, and they add an EGFR TKI in addition, then with the idea to do this double blockage for the external part of the receptors and the tyrosine kinase domain. Then it was a phase 1 presented with this drug and lazertinib, which is a third generation EGFR TKI in advanced EGFR mutant patients with two different settings. One settings of osimertinib resistance chemo-naive patients. Then at that time the patients exhausted all the available TKIs, but they also looked at treatment-naive cohorts. The dose escalation was in 26 patients, the dose expansion in 65 patients with sensitizing EGFR mutation, then the classical ones, and 20 patients were treated as naive patients when 45 was treated when they resist to the already available EGFR TKI. We have the results presented and illustrated on the right in these two different cohorts, was a response rate of 36% in osimertinib resistant chemo-naive patients with one complete response. And for the patient with, who were treatment naive, the response rate was 100% with a 20 partial response on the 20 patients treated in this cohort. And the median follow-up was quite short in both cohorts regarding what we could expect with targeted agents in these patients' populations. There was no dose-limiting toxicities identified during the dose escalation. The discontinuation rate was quite low. If we look globally at the adverse event, it was noted in 34% of the patients. 24% of the patients needed a dose reduction. And you can see the very good responses seen on the right, especially in the treatment-naive cohorts. It's a small cohort, only 20 patients. We should keep in mind that the response rate, the expected response rate with third-generation agents is around 85 to 90%. Then it could be better, but the sample size does not allow to a firm conclusion. What is a little bit surprising when looking at the osimertinib resistant cohorts is there was no report of the biomarker programs for those patients that we don't know if the mechanism of resistance was linked to a MET amplifications or not. If the mechanism of resistance was on target or off target, then if there was an interest to double block the EGFR pathway, or if the mechanism of resistance was completely different, then probably the interest of this double blockage was probably not as important. But it provides with another, I would say, targeted possibilities in these patient populations. However, we have to keep in mind that the response rate for the chemotherapy in these patients, in these pre-treated patient populations, could be up to 45–50% of the patients with a combination of chemotherapy. The place of immunotherapy in this patient population is also something we have to keep in mind. Then for the moment, it could be used in combination with chemotherapy. Then I don't know where will be the place of these agents. Probably we would love to see the biomarkers, and I can imagine that they have a large liquid biopsies program in order to see what were the both in resisting patients, the mechanism of resistance, but also in treatment naive patients, that if there could be a sign of concomitant alteration or sign of an increase in the MET, how they control the MET pathway, and of course we will see what will be the mechanism of progression in this patient's population. Then I would say both the design of the drugs is very interesting, and we know that double blocking EGFR and MET is something that we will probably have in our hand in the future. But once again, the prediction of the best setting to use this drug still has to be defined.
Great, thank you so much for that, Dr. Berlays. I think you really teed up some of the key sort of caveats and questions that come to mind with this data set around the resistance pathways and all of that. It'll be very interesting to see how this looks moving forward, especially with some of that translational analyses. We did have a few questions teed up here, but honestly, I think we really did hit all three of these questions along the way. So maybe in the interest of time, we'll just move right on ahead until our next topic here, which this topic was novel targets. Now we're going to go into novel technology. So the kind of protein engineering approaches, fusion proteins, and bispecifics. So we're going to first start off here with the interesting IL-2 based approach. Of course, IL-2 is something that has been known about for quite some time, developed at the NCI into a cancer therapy in the 1990s. But here we're talking about a new generation of IL-2 therapeutics. This is an IL-2 actually kind of fused to the alpha receptor of IL-2 in order to get more of that T effector versus T regulatory activity. And Professor Berlays, why don't you walk us through a little bit of the data that we saw here?
No, I think it's very interesting, and I'm not a specialist of the IL-2 drugs, but I was really interested to see how they design these novel agents. Just for the attendees, the idea was to say that when you try to target IL-2, in fact, you need very high dose of IL-2 in order to block the receptors. But when using these eye drugs of IL-2, then you will increase you will also target the alpha part of the receptors, and then you will increase the amount of Tregs. And then the idea when designing these drugs was to be able to target the beta delta part of the receptors, but not activating the alpha part, and just increasing the CD8 positive T cells with this kind of approach. Then it's how the drug was designed. You can see that there was monotherapy. There were two different monotherapy cohorts in melanoma patients and in renal cell carcinoma patients. The response rate in melanoma patients was not as high, but the number of patients is very, very low, 6 patients. One partial response was seen, and in quite aggressive melanoma subtypes, the mucosal melanomas. And when we look at the duration of response, it seems that in at least this patient, there was a quite prolonged response. They developed also the drugs in a combination with immune checkpoint inhibitors, and in this case, the pembrolizumab. You can see on the right the efficacy seen in these different cohorts of patients. In ovarian cancer, there was 1 complete response and 2 partial response out of 14 patients treated. Some additional responses were seen in triple-negative breast cancer, pancreatic cancers, and 2 esophageal cancer patients. The drug was quite well tolerated, 46% of grade 3 adverse event globally, but if we look at the treatment-related adverse events, the main adverse event was lymphocyte decrease and anemia and fatigue. 11% of those treatment-related adverse events led to a discontinuation of the drugs. There was no increase when combining the drugs with immune checkpoint inhibitors. And in the vast majority of the adverse events that were seen, if we look globally at the adverse events, they were related to an immune infusion reaction. One small drawback for this approach is the fact that the patients need to be infused 5 consecutive days over a 3 weeks period, which makes this approach, if it works very well, of course, interesting, but it needs to be compared to the other kind of approach we have when using these drugs. But I think that the mechanism of action is very interesting, and it's an approach we'll have to follow to see how it will work, and also to identify the patient population who will beneficiate the most from this approach.
Exactly, yeah, and I thought it was interesting that they were seeing some responses in some tumor types that you might not typically expect. There was a ovarian cancer, triple-negative breast, a pancreatic, a couple of esophageal. I mean, these are typically we would think of as like a cold tumor type. So kind of interesting to see. It kind of makes you wonder: is the IL-2 kind of getting something going with the T cells?
Well, these are small numbers of patients as well. I mean, the pancreas. I mean, you don't see responses in pancreatic cancer with IO therapy, typically, but you can get a little bit of response in the ovarian population. And so we'd probably have to, and we haven't seen these scans and we don't know a lot about these patients and their prior history. It is encouraging, but I think, it's early data. It's early data. I think the downfall is 5 days of infusion. Unless you have a gangbuster drug, it's hard to get compliance with 5 continuous days of dosing where the patient has to come in for 5 continuous days. But if you can get a decent response and you're not getting the same toxicity that IL-2 gave with the hypotension and the need for dopamine infusions, et cetera, I mean, that is encouraging. I mean, the safety profile it appears to be at least better in this trial than typical, IL-2 administration.
Yeah, we'll have to see how this data looks moving forward. I know this company is also exploring a subcutaneous approach, so that will hopefully lessen the burden. And then there's a couple of pegylated approaches in the clinic as well. So we'll see. I think the IL-2, this new generation of IL-2, is something that we have to see. How it's going to shape up with a few different approaches, some interesting tumor types, and data will tell. Okay. So the last kind of topic we want to discuss on in this kind of novel technology segment that we're here is just this broader idea of bispecifics, and maybe the data itself. We don't need to kind of dive into individually. These are all very early stage data sets, but 3 kind of quite different and novel approaches of bispecifics. One looking at 2 immune targets, one looking at more of a T cell engaging technique with PSMA. Another looking at a tumor antigen and co-stimulatory 4-1BB approach. Would be interesting to just hear from both of you your perspective on these kind of diversity of bispecific approaches and sort of what you see from this class overall.
Some people really like do you want me to start, Fabrice?
Yeah, sure. Please go ahead.
I mean, some people really like bispecifics, other people don't like bispecifics. I think that one of the things that we have to think about when we're talking about bispecifics are what are the two drugs that are making up the bispecific, or what are the two targets that are components of bispecifics. So if you look at the PRS-343 compound, it's HER2 4-1BB. You know, preclinically, 4-1BB has been an exciting target, but as a monoclonal antibody, you know, two 4-1BB targeted agents failed. Not because of efficacy, we don't know enough about efficacy, but because of significant hepatic toxicity, the BMS compound, urelumab, and there was one other compound, I forgot what company had created it. I don't know if it was Merck or who else. But if you can deliver, if you can use bispecifics to help deliver a drug or a targeted agent like 4-1BB that can be delivered in a bispecific such that you can overcome some of the toxicities of the previous singly targeted agent like 4-1BB, I mean, that's pretty exciting. Or if you can do an agonist-antagonist approach in a bispecific that can also minimize toxicity but enhance efficacy because you're delivering the drug you're delivering those two targeted agents simultaneously, it may be pretty exciting. And if you look at the Amgen 160 compound, you know, the PSMA CD3 bispecific, I mean, that data looks pretty interesting. There was a you know, I mean, I know this is PSA response, but there was also some RECIST response in this drug. PD-1, CTLA-4, maybe it helps overcome some of the toxicities of CTLA-4, and potentially you can get the dual efficacy of these two targeted drugs. You know, that may be something to you know, to consider. It's not only the efficacy. Efficacy is very important, but so is toxicity, especially if you haven't been able to deliver these targets or targeted agents because of the safety profile previously. I don't know, Fabrice, what do you think about bispecifics? I personally am encouraged by some bispecifics, but like I said, it depends on what the targets are that are included.
Well, I think it's the concept is interesting, and I know that people developing antibodies are looking at bispecific, three-specific. Then they say that they can do everything you want if you have the good target identified. Then I think the technology is something that is interesting, but is not the goal we are following for our patients. Then I think when we look at the PD-1 CTLA-4 bispecific antibodies, I think a setting that everybody can understand. I think that we even don't know, maybe with the exception of melanoma patients, where we have to use this kind of combination. Then I think at the end, if you are using two different drugs or one bispecific antibody, at the end, if we don't know what kind of patient we should treat and what kind of patient derives a benefit from these drugs, then I think at the end, the technology does not matter. Then it's why I think it's interesting to see that we can do that probably in the future, and it will be especially important when looking at immunotherapy, because we know that probably targeting one checkpoint will be probably not enough in some of the patients. Then probably we will need to target two different targets. Then having bispecific in our hand will be interesting. But as you said, we have also to face I would say the same kind of profile of safety, of toxicity profile compared to what we get when we use those, when we use drugs targeting those targets separately. Then I think it's another way. Maybe it could be a simple way to treat the patient, but it's not completely sure, and we need to be sure that we will use these drugs adequately. But the concept is interesting, and we had some preliminary interesting results, but we need to go ahead with the development of those drugs. But once again, the assessment of the targets and the assessment of the pharmacodynamics will be very important to be sure that we are doing what we are expecting to do.
Great comment. So maybe one that I'll ask on this side of the questions that we had teed up ahead of time. I think we kind of hit that middle one a little bit about toxicity. Maybe just this last one, toxicity on that, the PSMA CD3 compound, it had something around 25%, I think it was, grade 3 CRS.
Yes.
Will that be something that's a class-specific thing with bispecifics? Will there be kind of specific toxicity or administration hurdles that, as more bispecifics, especially in solid tumors, are coming through the pipeline, clinicians will need to familiarize themselves with?
I can start with that. So, I mean, you do see CRS with some of the bispecific, in some patients with bispecifics, but, you know, usually when you do see it subsequent to the, you know, administration time point where you did see it, a lot of times you can pre-medicate them to circumvent it with subsequent infusions. Other times you might not be able to. But, you know, with a lot of novel drugs, we see unique, especially when we were initially working in the IO landscape in the early days of IO therapy, we saw a lot of toxicities that we weren't used to seeing with cytotoxics. But as we started developing them and started seeing that they were effective agents and meaningful agents in certain tumor types and certain patient populations, we've started looking at the science of treating the toxicities and how to overcome these toxicities so that we can maximize the utilization of the drugs. You know, I don't necessarily know that, you know, CRS and the bispecifics alone as the only negative, if that was the only issue, would prevent us from moving forward with bispecifics. I think we can learn how to overcome it in several patients that manifest as CRS, because not everybody does. And again, it's bispecific dependent. But I don't know, Fabrice, what do you think?
Well, that's true that when developing these drugs, we had to prevent and to cope with this adverse event that seems to be unusual regarding what our experience with other kind of drugs in the past. But I agree that it's something that we could be organized to detect it as soon as possible and to manage as soon as possible, even if it's not so simple if we look at a large use of these drugs. For the moment, the use of these drugs is restricted to early phase development department, and I think we are organized to face this kind of side effects. We need to carefully look at how they will be used in the future if their use is larger and if there are some approvals for those drugs. But we are not there for the moment.
Great. Thank you both for the comments there. So we're just about the end of time. So, you know, we had a great discussion going across, starting at TIGIT, ADCs, novel targets like KRAS, novel technology like bispecifics, really robust. We covered a lot of ground. I hope we helped the folks who dialed in today to kind of understand what's coming in this next wave of oncology care. Maybe I'll pass it to each of you for a minute of just closing remarks and thoughts on this to close us out. Maybe starting with you, Dr. Borlesi, and then Dr. LaRusso, just your kind of closing remarks, and then we'll end the session here.
Well, I think we are living in a very exciting time. We have we understand the different diseases better and better. We have the drug to try to overcome the mechanism of progression to the currently available standards. Then it's really exciting. We also have a space to develop these new drugs in the early stages of the disease, which is also something very important, I believe. Then I think we could be confident that the progresses we are doing are providing the patients will provide the patients with better outcome very soon.
Yeah, I mean, I concur. I did get a couple questions. I can answer them quickly. One of them was about brain mets with the small molecule KRAS G12C and a comparator. So if you do have a drug like the Mirati compound, as Andrew, I'm referring to one of your questions, if one of these compounds has greater brain penetrance and can impact more so on brain mets than another one of these small molecules with equal efficacy outside of the brain, that is going to potentially be a deal breaker. I mean, I think one of the reasons why osimertinib quickly moved to front line in EGFR mutant non-small cell lung cancer patients or patients with that type of disease is because osimertinib did demonstrate efficacy in brain mets in the earlier trials, and it advanced forward and demonstrated that. So if you have a small molecule like a KRAS G12C targeted agent that has greater efficacy in brain mets than other agents of the class, honestly, it is going to, I think, be one of the deal breakers to move that drug forward.
Got it. Great. Thank you both so much for being so generous with your time, going through all of these different topics. I know it's not an easy task to bounce around between all of these things over the course of our 90-minute session, but I think this was absolutely terrific. For any of the audience members who are still on the line here, we have 4 more of these town halls planned. One actually just in 28 minutes on GI tumors with Dr. Kasi from Iowa, Dr. Catenacci from UChicago. Later this evening, we'll have Roy Herbst, Dr. LaRusso's colleague at Yale, and Suresh Ramalingam from Emory. And then tomorrow we have 2 wonderful panels on women's cancer with Dr. Beer and Dr. Brugo, really 2 of the preeminent figures in their respective spaces. And then GU tumors shortly after, tomorrow afternoon, with Dr. Srinivas from Stanford, Dr. McDermott from Boston, Thomas Powles from the UK. We'll go through prostate, bladder, and RCC, their respective areas there. So thank you everyone for tuning in today for the first of our town hall series, and we hope all of our audience members will tune in for the rest of the exciting program that we have. And then, of course, above all, thank you so much, Dr. LaRusso, Dr. Barlesi, for being so kind and generous with your time and engaging in this amazing discussion.
Thank you. Thank you, everyone.
Bye-bye.